All right, guys, so I'm going to work you through part of this RNA and protein synthesis digital interactive. So first thing is click on the link and then select File, Download, Microsoft PowerPoint. Once that downloads, go ahead and open it up. Make sure you enable editing. Okay, we're going to put our name on the front. All right, and it's same as what we've done before. So you're going to start, and you can use your notes to do this, comparing DNA to RNA, and then in the center, of course, is things they both have. For instance, they both have a sugar and phosphate backbone. All right, some of these bases are going to be found in both of them, like adenine is found in both of them, and some are unique to those. So you can use your notes to finish that one up, but there's a couple answers right there. Then we got pictures of the different kinds of RNA. So this straight one is your mRNA, and yes, the M is lowercase and the RNA is capital. This little squiggly thing is actually your ribosome, so it is rRNA, the R for ribosomal. And this one, it looks like it has a T in the center. All right, this is your transfer RNA, or tRNA. So messenger, ribosomal, transfer. All right, their functions are right inside your notes, so I'm going to leave that to you to do. You can just follow the note sheet, and it will tell you what each of them are. Down here we have transcription. So for genes to become proteins, DNA must first be, well, that's transcribed because we're talking about transcription. During this process, the enzyme, well, if we're doing transcription, we're not making DNA, we're making RNA. So we're going to be using RNA polymerase. The enzyme RNA polymerase binds to a section of DNA and assembles the nucleotides guanine, cytosine, adenine, and now remember we're making RNA here. So what base does RNA have that DNA does not? Uracil. Okay, so, and uracil into a, well, RNA is single-stranded, where DNA is double-stranded. That's why it's a double helix. This is a single helix. All right, down below we have our base pair rule, a little practice with that. So remember, we're going from DNA to RNA. So A can't go with T anymore. T's not even an option. And instead, you're going to have U for uracil. Cars still go in the garage. So C and G go together. C and G go together. And then if I have a T, the other side can be A, but you just can't have the T's in RNA. All right, so then we're going to compare replication during versus transcription. So remember, replication is the process of making DNA. Transcription is the process of making mRNA. So if we're doing DNA, we're doing this before our cells divide. Okay? And we're making DNA. If we're transcribing it, we're just reading our DNA and making a copy of it. So I'm going to have you guys finish the rest of this using your notes, but that should get you started. Here you're going to be dragging your bases again going from DNA to RNA, so you'll notice there are no T's in here. You can drag that around and do it because we just did an example. This is the same, just a little more practice. This one I'm going to do with you. This one says something that happens with our mRNA, and I don't usually teach this part, but because we are talking about COVID and we have an mRNA vaccine, and a lot of people don't understand how that works. So I wanted to go ahead and show you an extra step that occurs there. I will not test you on this. You just have to do it, okay? So in our mRNA, we have introns, which are sections that do not code for proteins. And then we have exons, which are sections that do. Well, the exons are called that because these are the parts that are going to actually exit the nucleus, and the introns don't. Okay, because they're not helpful. They're just what we call junk DNA in this area, copies of the junk DNA. So what our bodies do is they use enzymes to cut these little snippets out before it leaves the nucleus. So if we look at our instructions, it says remove the introns, and introns are red, from the pre-mRNA, they call it pre-mRNA because it's not done, and splice together the remaining exons to produce the final mRNA. So this is something they had to do to make our COVID vaccines. So we're going to take the red out, and we're going to take our blue, because this is the part that actually codes for genes. And in the case of COVID, it's coding for the genes for a protein on the surface of the virus. So it doesn't code for the whole virus, just the little part of it that your immune system needs to have an immune response. So all this junk part will be cut out, and this is the important gene coding part there that will exit our cell. Okay? So proteins are made by joining the monomer of a protein, which hopefully you remember the monomer is amino acids, into long chains called polypeptides. Because remember, poly is many, P in peptides is for protein. So that's many proteins, long chains. All right, a blank consists of blank consecutive mRNA nucleotides. So when we have 3 messenger RNA codon nucleotides, I said the answer a minute ago, it's a codon, and we do indeed read that 3 letters at a time, as you could tell by the practice sheet that you guys did. All right, so a codon consists of 3 consecutive mRNA nucleotides that code for a specific amino acid. So that's where we use our chart. There are 64 possible codons. Now, that doesn't mean there's 64 amino acids. There's not. There's only 20, because if you go back and look at your chart, a lot of them repeat, okay? So there's 64 possible codons. I don't expect you to know that number. I do expect you to know that one. All right, the codon blank serves as a start. That's going to be AUG, as you could tell from your worksheet you did earlier, that gives you MET. Serves as a start codon for your amino acid sequence, or the promoter. There are also 3 codons, and you can put these in any order, it doesn't matter. All of these, if you look at your chart, code for stop. All right, and the stop codon tells it to stop making that protein chain at that point. If you need a little help still with your chart, here's a video here that you can watch to show you how to read the charts. You're going to be reading that chart to find the amino acids. So you're going to be writing down these letters in here, even if it's stop, just like we did on our practice sheet. So you got 2 slides of that. All right, then down here we have a picture, and this is somewhere kids have a hard time because these pictures of transcription and translation are so busy. So down here I have this long skinny chain that's usually the easiest to identify right away. That was copied from the DNA. That is your mRNA. All right. Then this bubble is pointing to 3 letters on the mRNA. So those 3 letters are called a codon. Codon. The opposite 3 letters that are on this molecule, or the opposite of a codon, is an anticodon. There we go. So this big blob that everything is at is an organelle. That organelle is the ribosome that it goes to, because ribosomes make proteins. That's what we learned when we did organelles. This molecule that is T-shaped in the middle and has the anticodons in it that also have a T, that is your tRNA. Up here, your tRNA carries these molecules. These molecules are your amino acids that they transfer around. And when you take many amino acids and they polypeptide together, that is your growing protein. All right, so all that is what occurs in translation. Remember, prior to this, transcription occurred where you copied your DNA code into the mRNA. Down here, we're going to put these in order, which is a little bit tricky. So we're going to grab and try to separate first what happens at the end and the beginning. So when we're talking about the ribosome, that automatically is going to be translation. So anything with the ribosome, translation. Now, when our introns are moved, which we just practiced, that's up here. That's going to be part of translation. RNA polymerase transcribes, it's got something to do with DNA. That's got to be part of transcription. Transfer RNA, bringing things to the ribosomes, that's going to be translation. And amino acids, when you're making that protein, that's going to be translation. So now we got to figure out which one of these is first. Do we transcribe the RNA based on the DNA first or get rid of the introns? Well, you got to do the actual transcription before you can get rid of the introns. So this kind of ugly orangey brown one is first, and then this yellow one will be second. All right, so that is transcription. Now we're at translation. So the first thing that happens in translation is your mRNA docks at the ribosomes. So we're not making amino acids right away. We can't bring our tRNAs in right away. Oops, I made a shift over on you. But the ribosome attaches to the mRNA, yes. That has to happen after the mRNA leaves the nucleus. And then we have to start the process. So we're not going to be stopping it. We got to start it first. So transfer RNA is going to start bringing the amino acids, right? And matching up by complementary codes. And then the amino acids continue to join, forming those polypeptide bonds. And then when the ribosome reaches a stop codon, then our protein, our polypeptide, will be released, and that's the end, okay? Then you guys have to do your code again. Remember to flip. Remember there's no T's in mRNA. And find your amino acid code. You could go back to slide 15 to use the chart up here if you want, or if you click on this, you could do split screen and have your chart up while you're doing this. So you have 2 slides of that to complete. Hope that helped, guys. Good luck. Bye.